Hello nerds, welcome to MedExpert. If you're looking for reliable, evidence-based medical content, you're in the right place. We provide expert insights, health tips, and the latest medical updates. In the previous parts, we explored some organelles. If you haven't watched those yet, make sure to check them out. In this third part of our cell organelle series, we'll explore the remaining organelles, starting with Plastids' powerhouse of photosynthesis. Plastids are double-membraned organelles found in plant cells and certain protists, like algae. They contain pigments and play a crucial role in photosynthesis, storage, and pigment synthesis. Plastids can be classified based on the pigments they contain or their developmental stages. Based on developmental stages, plastids are classified into two types: proplastids and mature plastids. Proplastids are immature, young plastids found in meristematic cells. They are self-replicating organelles and can differentiate into chloroplasts, chromoplasts, or leucoplasts based on cellular requirements. Based on the type of pigments they contain, plastids are classified into three main types: chloroplasts, chromoplasts, and leucoplasts. Let's explore the unique characteristics of each. Chloroplasts. They contain chlorophyll and other pigments that capture light energy to produce glucose and oxygen through photosynthesis. Under a light microscope, chloroplasts appear as multiple granules within the matrix. These granules, called grana, are stacks of thylakoids containing chlorophyll, the key pigment for photosynthesis. Under an electron microscope, three main structures of a chloroplast are observed: envelope, stroma, thylakoid. Envelope is a double membrane consisting of an outer and inner membrane that encloses the chloroplast. Stroma, the fluid-filled matrix containing enzymes, DNA, ribosomes, and starch granules. The chloroplast, especially stroma, is the site of CO2 fixation during the Calvin cycle, where carbon dioxide is converted into sugars through the process of photosynthesis. Thylakoid is a network of flattened, membrane-bound sacs, thylakoids, arranged in stacks called grana. The membranes of the granum play a key role in light absorption and ATP synthesis, as they contain chlorophyll and essential proteins for the light-dependent reactions. Each granum is connected to others through a non-green structure known as the intergranum or stroma lamellae. This connection helps in the transfer of molecules during photosynthesis. Chromoplasts are specialized plastids that provide colors to plant parts other than green. They are found in petals, fruit walls, and other brightly colored structures. They contain carotenoid pigments, such as carotene and xanthophylls, which are responsible for their vibrant hues. These pigments play a crucial role in pollination, as they help attract insects, aiding in the reproductive process of plants. Leucoplasts are colorless plastids, primarily found in the parenchyma cells of roots, stems, and seeds. Their main function is storage, and they are classified based on the type of biomolecule they store. Based on the type of stored compound, they are classified as: amyloplasts store starch, commonly found in tubers and seeds. Eleoplasts store lipids, especially in oil-rich seeds. Proteinoplasts store proteins, which are abundant in legumes and nuts. These plastids play a vital role in plant metabolism and energy storage. Centrioles are cylindrical, non-membranous, self-replicating organelles found in animal cells and lower plants. Centrioles are positioned at each pole of the cell, near the nucleus. Structurally, each centriole is composed of 9 triplets of microtubules arranged in a circular pattern. These organelles are essential for spindle fiber formation, ensuring proper chromosome separation during cell division. Centrioles also give rise to the basal bodies of cilia and flagella. These basal bodies anchor the cilia and flagella to the cell membrane, enabling their formation and function in cell movement and signaling. Presence of cell nucleus was reported in 1831 by Robert Brown. The nucleus is a membrane-bound organelle found in eukaryotic cells, housing the cell's genetic material. It regulates critical cellular functions like growth, metabolism, and reproduction. The nucleus is surrounded by a protective nuclear envelope and contains the nucleolus, which plays a key role in ribosome production. The nucleus is usually a large, prominent structure located in the center of animal cells, except in muscle cells, where it is positioned at the periphery. In plant cells, the nucleus is often pushed to the periphery due to the presence of a large central vacuole. The nucleus can vary in shape, being either irregular or spherical. Typically, cells have one nucleus, known as mononucleate. Cells containing two nuclei are referred to as binucleate, while those with more than two nuclei are called multinucleate. The nucleus is visible only when the cell is in its non-dividing stage. It contains a chromatin network and a soluble sap known as nucleoplasm. During cell division, the nucleus disappears and the chromatin condenses into chromosomes. These chromosomes carry the hereditary material, which regulates and controls all the activities of the cell. The chemical composition of the nucleus includes DNA, RNA, and proteins such as enzymes. The nucleus consists of the following components: the nuclear membrane, which encloses the nucleus and regulates the movement of materials; the nucleolus, responsible for ribosome production; the nucleoplasm, a semi-fluid substance inside the nucleus; and the chromosomes, which carry the genetic material in the form of DNA. The nuclear membrane is a double-layered membrane that surrounds the nucleus, isolating it from the cytoplasm. It consists of two layers, an inner membrane and an outer membrane, with a space between them known as the perinuclear space. The nuclear membrane consists of an outer and inner membrane. At specific points, these membranes merge, forming nuclear pores. These pores serve as gateways, regulating the exchange of materials between the nucleus and the cytoplasm. The number of nuclear pores varies depending on the type of cell. Undifferentiated cells, like eggs, have a large number of pores, about 30,000 per nucleus. In contrast, differentiated cells, such as erythrocytes, have only 3 or 4 nuclear pores per nucleus. The nucleoplasm is a transparent, semi-fluid substance inside the nucleus, composed of a variety of components. It includes proteins, enzymes, RNA, DNA polymerases, free nucleotides, and essential metals, such as magnesium, which are crucial for DNA synthesis. Additionally, it contains histone and non-histone proteins, which play roles in DNA packaging and regulation. The nucleolus is a dense, spherical structure located within the nucleus. Unlike other organelles, it lacks a membrane and is made up of RNA, proteins, and DNA. Its primary function is to synthesize ribosomal RNA and assemble ribosomes, which are essential for protein production. Cells with high protein synthesis activity, such as liver and nerve cells, have a large and prominent nucleolus. The nucleolus may be single or multiple within a nucleus. It appears during interphase but disappears during cell division. Structurally, it has two distinct regions: an inner granular area, rich in ribosomal RNA, and an inner fibrillar core, containing ribosomal DNA, which is essential for ribosomal RNA synthesis and ribosome production. Chromatin is the complex of DNA, proteins, and RNA that makes up chromosomes within the nucleus of eukaryotic cells. It plays a crucial role in gene regulation, DNA replication, and cell division by packaging DNA into a more compact structure. Chromatin exists in two forms: euchromatin and heterochromatin. Euchromatin is loosely packed and transcriptionally active. This means genes can be expressed. On the other hand, heterochromatin is densely packed and transcriptionally inactive, meaning genes are silenced. In simple terms, euchromatin is like an open book, easy to read, while heterochromatin is tightly shut, keeping its contents hidden. The nucleus is deeply stained with basic dyes because of the presence of chromatin material. During cell division, this chromatin condenses into thread-like structures known as chromosomes, which appear darkly stained under the microscope. Chromosomes consist of two chromatids joined at a specialized region called the centromere, which lies within the primary constriction. Some chromosomes also have secondary constrictions, known as nuclear organizers, which contribute to nucleolus formation during interphase. Each centromere contains a complex protein structure called the kinetochore, which plays a crucial role in microtubule attachment during cell division. Chromosomes are classified based on the position of their centromere. Metacentric chromosomes have a centrally located centromere, giving them equal arm lengths. Submetacentric chromosomes have a slightly off-center centromere, resulting in one shorter arm. Acrocentric chromosomes have the centromere near one end, making one arm much shorter than the other. Lastly, telocentric chromosomes have the centromere at the very end, forming a single visible arm. Peroxisomes are single, membrane-bound organelles involved in metabolism and detoxification. Christian de Duvet discovered peroxisomes by isolating particles from liver cells and other tissues. These organelles were found to be enriched with oxidative enzymes, including peroxidase, catalase, glycolic acid oxidase, and several others, which play a crucial role in cellular metabolism and detoxification. The term peroxisome was given because this organelle plays a key role in the formation and decomposition of hydrogen peroxide within the cell. Peroxisomes contain the enzyme catalase, which breaks down hydrogen peroxide into water and oxygen, preventing potential cellular damage from oxidative stress. Glyoxisomes are specialized organelles found in plants that contain enzymes such as glycolic acid oxidase and catalase, as well as several unique enzymes not present in animal cells. These organelles are involved in processes like photorespiration and lipid metabolism, playing a key role in seed germination and providing energy for early growth. Glyoxisomes are present only during a short period in the germination of lipid-rich seeds, where they help convert stored lipids into carbohydrates. These organelles are absent in lipid-poor seeds, such as peas, as those seeds do not rely on this metabolic process for germination. One of the primary activities in germinating seedlings is the conversion of stored fatty acids into carbohydrates, which is essential for early growth. This process is achieved through the glyoxylate cycle, with the necessary enzymes located in the glyoxisomes. The cytoskeleton is a dynamic network of protein filaments and microtubules that gives the cell its shape and structural support. It is involved in key processes like cell division, intracellular transport, and cell movement. The cytoskeleton consists of three main components: microfilaments, intermediate filaments, and microtubules. The main proteins that make up the cytoskeleton include tubulin, which forms microtubules, and actin, myosin, and tropomyosin, which are also found in muscle cells. These proteins are essential for maintaining cell structure, enabling cell movement, and supporting muscle contraction. Microtubules are hollow cylindrical structures composed of tubulin proteins. They serve as the cell's internal framework, providing structural support and facilitating intracellular transport. Microtubules are also essential for cell division and form specialized structures, such as centrioles, cilia, and flagella, which aid in movement and organization within the cell. Microfilaments are thin, flexible protein filaments composed mainly of actin. They help maintain cell shape, provide mechanical support, and enable cell movement. These structures are essential for processes like cytokinesis, muscle contraction, and intracellular transport, ensuring proper cell function and organization. Intermediate filaments are cytoskeletal components with a diameter of 8 to 10 nanometers, making them intermediate in size between microtubules and microfilaments. Their basic protein subunit is vimentin, and each filament consists of 3 twisted chains, forming a hollow space. These filaments create a supportive network in the cytoplasm, providing mechanical stability and maintaining the cell's shape. And that wraps up part 3 of our series on cell organelles. With this, we've completed the structure and function of the cell. If you haven't watched the previous parts, make sure to check them out. And don't forget to subscribe so you never miss an update. See you in the next video.